Hi, I'm Marnie Hodge. I'm the Land Plan Development Officer at Sheep Genetics, and I'll be running through Analysis Enhancement 2, which is using new data and the improved analysis of carcass and eating quality traits in the genetic evaluation. We'll be looking at using eating quality data that's collected in plant, as well as updates to the shear force validation and transforming carcass data to a standard contemporary group mean. If we focus firstly on using eating quality data that's collected in plant, we know that meat science phenotypes are very expensive and time consuming to measure. And across Australia, we've got more and more processors with the capability to collect eating quality data in plant, which is really great because this means a faster and cheaper way to collect this data objectively. So we have two data types that are being collected in plant at the moment, which is IMF or intramuscular fat and lean meat yield. And this can be collected via an MEQ probe or SOMA device for IMF or via a DEXA machine for lean meat yield. So as of the 15th of May, analyses runs for Land Plan and the 21st of May runs for Merino Select. We'll be having about 1,500 phenotypes for IMF added into the analysis, as well as about 4,500 phenotypes for lean meat yield added into the analysis. So if we focus first on IMF, we know that the gold standard is collection of chemical IMF, and this is done on every research flock animal. But we do know that we can collect IMF through a MEQ probe, which is pictured on the top, and this can be done at chain speed. We can also collect IMF via a SOMA device, which requires a cut surface but can still be collected in plant. Now, if we have a look at this graph here, the x-axis has transformed chemical IMF breeding values, and that's plotted against breeding values using all three data types for IMF. And we can see a really strong genetic correlation here. Moving on to lean meat yield, we know that the gold standard is collection via a CT machine or computer tomography machine, which is pictured below. But we can also collect lean meat yield data via a DEXA device, which collects lean muscle, bone, and fat, and this can be used to inform lean meat yield and dressing breeding values. Again, with this graph, we've got the gold standard, which is CT lean meat yield breeding values plotted on the x-axis against breeding values for both CT lean meat yield and DEXA. And we again see a very high genetic correlation between these two. So if we hone in a little bit more and have a look at the changes, this graph is for IMF and lean meat yield just in the terminal analyses. So what we've got here are breeding values for IMF and lean meat yield where an animal does not have a genotype, which is plotted in blue, where an animal has a genotype, which is plotted in green, and where an animal has gained a genotype, which is plotted in orange. So Kate will talk a little bit later about our updates to our genomic reference. So keep this in mind for that section, but essentially we can see a little bit of movement with the addition of more information into the analysis. So this graph on the right here again has IMF and lean meat yield, and this is changes to breeding values in the terminal analysis again based on the presence of data. So where there has been pre-existing data in the analysis versus where there's been addition of new information essentially through the collection of MEQ and SOMA IMF as well as DEXA lean meat yield. So if we focus first on IMF, which is on the left, we see a really high genetic correlation of about 0.985 between where an animal has had existing data versus where there's been new data added into the analyses. And then on the right, there's the lean meat yield analyses plotted for terminals, which is, again, a high genetic correlation, but it's a little bit lower. It's at about 0.652. Which is likely because there's more data points that are being added into the analysis for lean meat yield compared to IMF. So some considerations if you're thinking about consigning animals to a processor that collects eating quality data in plant. We know that hook tracking is still a very big issue, so we need to make sure that the data that's being captured is accurate before it flows through to the genetic evaluation. So prior to consignment, please contact Sheep Genetics as well as the processor and inform the processor that you are consigning animals for kill and that kill data is going to be used in the genetic evaluation. So we can keep a bit more of a close eye on hook tracking. We also are going to need to take a pre-slaughter weight as well as the date of measurement of that weight in the management group, just like any other on-farm phenotypes. And we, so Sheep Genetics and AGBU will use this pre-slaughter information and essentially line it up with the carcass data that's provided to you post-kill to ensure that we have accurate data that's being utilised in the evaluation. So it's also important to note that we don't currently have a way to capture shear force in plant at the moment. So we still need to be collecting phenotypes for shear force data. So it's still very important to nominate size to the resource flock to capture shear force phenotypes. So update 2 in this section is the update to the shear force validation. So as part of this larger body of work, AGBU also re-estimated the shear force phenotypes and identified that there was some phenotypes that had a contemporary group mean of greater than 50 Newton meters. So where a contemporary group mean had a mean of greater than 50 Newton meters, this data has been excluded, and it's been identified that there was non-genetic impacts on performance for this particular range of data where the group mean was greater than 50 Newton meters. So there's only about 800 phenotypes that are actually being excluded from the analysis, which means that there's still about 20,000 phenotypes that are being used in the analysis across both Land Plan and Merino Select. So as part of this improved filtering of outliers, where we were able to actually exclude data that has had a non-genetic impact on shear force phenotypes. And this has resulted in a better heritability of shear force. So the final analysis enhancement in this section is the transformation of carcass data to a standard contemporary group mean. It was identified that some contemporary groups had a non-normal distribution. And if we have a look at the top graph here, which is DEXA lean meat yield and the distribution of that, we can see that this isn't normal. The distribution is not normal. Where this same data has been transformed to a standard contemporary group mean, which is shown in the bottom graph, we can see that the data distribution is more normal. And this transformation of data to a standard contemporary group mean for carcass traits has helped to account for slight variances across technologies which capture carcass and eating quality data. This may result in movements in breeding values for lean meat yield, dressing percentage, IMF and shear force across both Land Plan and Merino Select, and obviously in indexes that have relevant carcass data in both Land Plan and Merino Select. So ultimately, what does this mean for you? We have more and more phenotypes which are used in the analysis, which is great because historically this carcass and eating quality data has been very expensive and time consuming to capture. So it's really great to be able to utilize more data in this sense. We also have better validation of carcass and eating quality data. However, this update may actually result in some movements in breeding values and indexes where there is a direct relative with phenotypes for eating quality or carcass data.